Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kame microbiologist. Today in this video lecture we are going to talk about the last stage of the eukaryotic translation, which we call data termination of the translation. So this is the fourth video lecture of this series, and in this video lecture we are going to talk about termination of the translation. Now in the last three video lecture of this series we talked about the protein synthesis overview and requirements for the protein synthesis, and after that we talked the initiation of the translation, and after that we talked about the elongation of the translation. Now this is the last video, and in this video we are going to talk about the termination of the translation in a detail. So please stay tuned and keep watching. Okay, so let me write eukaryotic termination of the translation. So again in this video we are going to talk about the introduction of the eukaryotic termination of the translation. We are going to talk about the requirement, we are going to talk about the steps involved in this case, and we are also going to talk about the mechanism of each steps. So first of all, let me talk about the eukaryotic termination of the translation. So it is the last stage of the translation in which the ribosomes will encounter the stop codon which is present on the messenger RNA, and after that we get a long polypeptide chain and it will be released out. After that this ribosome will also break down, and due to this way we get a long polypeptide chain and it will be further converted into a protein. So this is called as termination of the translation. So in this case what actually we required, so in this case we required all those things which we talked about in initiation of the translation and elongation of the translation. But moreover we also required the release factor. Release factors. And we will be denoting it by E or F. E means eukaryotic, R means release, F means factor. So eukaryotic release factor, and we required eukaryotic release factor 1, 3, and moreover we required eukaryotic release ribosomal factor, or we also called it eukaryotic release recycle or ribosome recycle factor as well. These are the three main factors, release factor, that we required in termination of the translation. But moreover we also required the stop codon. Like UAA, UAG, and UGA. So these are the stop codon. Now what happened? These are the main things that we required. So now let's talk about the steps. Now in this case, what kind of steps involved? Let me write here. So the first step we call that activation of release factors. This is the first stage. The second stage is cleavage or hydrolysis of polypeptide chain. And the last is breaking or breakage of ribosomes into subunit. Into two subunit. So these are the steps that are involved in termination of the translation. Now let's talk about each steps with the help of mechanisms. So let me write mechanisms. Now what happened? In the previous video, if you guys remember, we are at that stage. Let's suppose this is the messenger RNA, as you can see. Now this is the ribosomes. Okay, so let's suppose this ribosomes will reach to the stop codon, which we called it UAA. So once the ribosome encountered this stop codon, so after that, let me draw the polypeptide chain here. So let's suppose this is the polypeptide chain. Okay, so now let's suppose this is the long polypeptide chain which is present at the P site, and now the E site is also filled with this uncharged transfer RNA, and A site is again free, as you can see. Now once this ribosome reached to this stop codon or encountered this stop codon, any one of the stop codon, it could be UAG, UGA or UAG. Once it reached, so then after that what happened, the first stage will be occur, which we called it activation of the release factor. Now in this case, the first release factor, which we call it release factor 1, will get activated. So release factor 1 is again have the same structure which we talked about in the prokaryote. They have again the 2 domain, as you can see. So its first domain is using for the hydrolysis, means it will break this polypeptide chain, while its other subunit is used for the binding to these sites. And remember, this is the release factor 1, eukaryotic release factor 1. Okay, it will be brought by this release factor 3. So it cannot be, directly move and bind to there. It will brought and it will be moved. The movement will be possible by this release factor 3. So what happened? Release factor 3 will bind here, as you can see. This is, let's suppose, release factor 3. Eukaryotic release factor 3 will bind here, and it will brought this release factor 1 to this A site. So after that it will bind here. So as you can see, now they have a kind of nucleotide which is complementary to this UAA. After that it will properly bind here. And what happened, again in this case we required the GTP, which is converted into GDP and inorganic phosphate. So energy is required for the breakage and moreover for the binding. Now it will break this hydrogen bond, as you can see, a polypeptide bond, not the hydrogen, or polypeptide bond between this transfer RNA and between this long polypeptide chain. So once it will break down, so after that what we get? We will get the chain, as you can see. So let's suppose this is the protein, as you can see. Now we will get a long chain of the protein. After that what happened, this is not the end of termination. After that what happened, a next release factor will be activated, what we call that ribosomal release factor or ribosomal recycle factor. It will get released. But moreover we also required, in this case we also required the elongation factor as well. Elongation factor 2 or G. And moreover we required the initiation factor as well, and initiation factor 1, 2A and 3. These 3 things are also required in this termination. Sorry, I forget to write here. Now what happened after that? After that, next release factor will get activated, which we call it ribosomal release factor or elongation factor 2. Moreover, elongation factor 2 will also get activated here. So the 2 things will get activated here and bind here. So the first is ribosomal release factor, while the second is elongation factor G or 2. It will get activated and bind to that specific area. Okay, now what happened? This ribosomal release factor once bind to that specific area, so they will, released out this release factor 1 as well as release factor 3. So release factor 1 and release factor 3 will be released out. RF1 and 3 will get released out by this ribosomal release factor. And due to this way, it will bind to this site, as you can see. So let's suppose this is the release factor 3, as you can see. This is the ribosomal release factor. Now once it bind here, they have also a kind of nucleotide which is complementary to this UAA or UGA. Once it bind to that specific area, so then what happened, again we have the two uncharged. If you remember, this transfer RNA is now uncharged because the polypeptide chain are now released out. And this is the previous one, uncharged transfer RNA, as you can see. What happened? This release, ribosomal release factor, will activate this elongation factor 2 or elongation factor G, eukaryotic elongation factor. Once it activate it, so what happened? It will be binded here or come here, and due to this way translocation, again the translocation will occur, because we talked about this EFG. The main function of EFG, they will help in the translocation, so the ribosomes will move. Now remember, when the ribosome a little bit, once it push, so what happened? It will break down. Just a little bit, the pushing, once a little bit the pushing will occur, it will break down, or this ribosome will be, break into two subunit, as you can see. After that, the ribosome will break down, and the uncharged transfer RNA, moreover this messenger RNA and the ribosome are, everyone thinks are get released out, and after that we will get the situation like that. So let's suppose this is the elongation factor G. Okay, now in this case again what happened, the GTP is required, which is converted into GDP plus inorganic phosphate. So now the situation will be like that. So this is the transfer RNA, as we can see. These are the large subunit, and let's suppose this is the small subunit, and these are the uncharged transfer RNA, as you can see. And this is the ribosomal release factor. Okay, after that what happened? 3 initiation factor, like initiation factor 1, 2A and 3 will bind to this small subunit, as we can see, to this small subunit of the ribosome at the E site. Means this, let's suppose these are the 3 initiation factor, like initiation factor 1, 2A and 3, it will bind to this E site. And moreover, initiation factor 1A will bind to this A side. So again, E side and A side are now blocked, as you can see. And what it will actually going to do, the main function of each initiation factor, they will prevent this large subunit to bind with the small subunit. So again, the binding will be prevented by this initiation factor, and this is how the termination of the translation will occur. And after that, this protein will be folded, but we will explain the folding inshallah in the next video lecture. This is all about the eukaryotic termination of the translation. Sometimes this folding process is also co-translation, and sometimes it will also the what we can say, after the translation this folding will also occur, but most of the time it will occur after the translation. Okay, and this is all about eukaryotic termination of translation. I hope you get idea about that, but if still you have any kind of question in your mind, you can write it into a comment. Inshallah, we'll give you answer as soon as possible. If you like these videos, so make sure to hit the like button on all these videos, and also subscribe the channel, and thank you so much for watching.